All right, we're going to look at the second part of our notes on genetics. So we're just beginning to learn the basics and much of what we know about genetics and how genotypes lead to phenotypes and how genes carry traits or alleles and how they're shared with offspring comes from studies from Gregor Mendel. So you were supposed to watch his the video about his research. If you do not have already, it would be really handy for you to have like a yellow and green for this part of the notes. So if you could just grab, you could pause this, grab colored pencils or highlighters or crayons or whatever, but yellow and green would be handy for you to have, okay? So Gregor Mendel was a monk, and so that is a religious, he had made a vow to be loyal to the church. You guys have probably heard of nuns, but monks are kind of the male equivalent. So he was a monk living in a monastery, but he, you know, we've talked before that you don't have to have a science degree to do studies and to do experiments and to think logically. So he was a monk, but he was really interested in studying the traits of plants. So he grew a lot of plants, and he was really interested to see what caused some plants to turn out with certain traits and some to turn out with others, and he felt like maybe that expanded beyond just plants. So he lived, or actually not lived, but he studied. He did several studies between the years of 1856 and 1863. So this was a long, long time ago that he did this work, and people have been building on it ever since. So his work was done in the late 1800s, and one of his famous studies you watched the video about was on Mendel's, it's called Mendel's pea plants. Okay, so Mendel had two different, he noticed that he had green peas and yellow peas, okay, and that's where your colors come in. And so he wanted to see why were some peas green and why were some peas yellow. And so, to make a long story short, he started with kind of a purebred green plant and a purebred yellow plant, and then he wanted to cross them and see what would happen. So in this case, in Mendel's, we're going to use a capital Y for yellow. Remember, we could use any letter that we want, but we just try to avoid letters that look very similar when they're lowercase and uppercase. So a capital Y for yellow, and then a lowercase y, which is easy to see because it's much smaller, is going to be green. Okay? So Mendel started out with a green, which we know can only be green if it has two recessive together. All right, otherwise the dominant trait will outweigh the other. And then he started with a homozygous recessive and a homozygous dominant. Okay, so you can kind of color those in. And he bred them together. And what he ended up with, when he combined them, he ended up with the offspring, and he worked in percentages, so we're just going to make this an easy number of percentages. What he ended up with was he knew that one allele had to come from the mother or father, and then one allele has to come from the other parent. So he knew that the combination here would be one allele from each. He knew there would be a yellow and a green in each of these because they had to receive from each parent. But what happened when they all came out, he didn't really know about dominant and recessive. He just knew that they all of the offspring came out yellow. So this was in his, they call this his first generation, the F1 generation, came out all yellow. And so like any good scientist, he asked, why were there no green? There was a green pea in there, a red and a crossed a green pea and a yellow pea, and all I got was yellow peas. And so then he had to kind of ask a little bit further. So then he went ahead and he mixed the second. So he bred 2 of the offspring. These are just plants, so offspring would not breed in humans. But he bred 2 of the offspring, and then something kind of surprising happened. Was that he bred 2 yellows together, and he did end up with 75% of the offspring were yellow, but then this little surprise came, and 25%, 1 out of 4, came out green. So that told him, that's when he started coming up with this system of, okay, I know that when these were mixed together, they're going to get one. This was his theory, and it has since been proven, that they're getting 1 allele from each parent. So in this case, they got 1 from each parent, but the yellow was dominant. But in this case, most of the time, they're going to end up with an allele, a dominant allele in there. But this guy, what happened was that one ended up getting both of the recessive alleles. They got the recessive from mother and recessive from father. So 2 recessive meant that pea came out green. Okay? So that's what he had to figure out. Where did this green come from? So other people have kind of looked at his research, they've looked at what he came up with, his theories, and they have found different ways to prove that he was correct in what he was thinking. And one of the people that started kind of looking at his work was this professor from Cambridge University in England. So his name was Professor Arthur Punnett. He had 2 last names, Balfour Punnett. And in 1912, he came up with what's called the Punnett square. And it's a way that you can figure out what's the likelihood that a cross between this and this will end up giving you each color or each trait. So you could do this with colors, you could do this with shapes, you could do this with sizes, tall, short, you could do it with the shape of your eyes, you could do it with the color of your eyes, your hair. There's all sorts of traits that we have, and so you can really calculate that. So he came up with something called a Punnett square, and a Punnett square, a basic Punnett square, is just 4 boxes. They can be bigger, but they're just basically 4 boxes. And so you take what you started with, so we're going to look at this second generation and see why this happened. You kind of take what you started with, and if we had dominant and recessive, well, I shouldn't have written that there. If we had, the parents were dominant and recessive, and then another dominant recessive, so 2 heterozygous, and we just split those parts. So we take the capital, the dominant, we put it here, we take the recessive, we put it here, ignore that guy. Then from the second parent we take the dominant and the recessive, and then basically you just work out your combinations. So if I took the dominant and dominant, I would end up with 2 together, so we know that that would be yellow. And then if I have the dominant and the recessive, put that combo in, recessive and dominant, and then recessive, recessive. So if I looked at this, when I look at the possible combinations, I see that 3 out of the 4 would still end up yellow, and only 1 of the 4 would end up green, just like Mendel figured out. So we use the Punnett square. The Punnett square is used to calculate probability, like what is the likelihood of a trait. So we could see that with this combination, 2 heterozygous, then we have 3 of the 4, which is 75%, probability of getting a yellow pea, and only 1 of the 4, which is 25%, probability of getting a green pea. Okay, so we could look at a few more of those examples, and we will in class. We're going to do some practice with Punnett squares, but just remember, you're just basically pulling each trait together and seeing what it would come up with. So you guys are going to have some Punnett square practice to see what the point is to calculate the probability of a trait showing itself, okay, of a phenotype being visible.